Thank you, and I'd like to thank all of you for being here and for the organizers for inviting me. We would love to actually be doing more transcriptome analysis, which is one of the reasons I came here, and in fact are beginning to do that. One of the problems we have is a lot of what we do is genetics of psychiatric diseases, and we just can't get the RNA. But cancer is a different story, and that's what I'm going to talk about today. I'm going to talk mostly about genomic analysis, though, but also some about transcriptome analysis, because this is very much ongoing work, and the transcriptome analysis is actually what's mostly ongoing. So, as I mentioned in my comments before we started, most of what we have been doing recently, in my lab as well as others, has been Illumina sequencing. And that's been an incredible burst of information has derived from that. When we first started trying to sequence the genome in 1990 or so, 89, those of us, I think, in the trenches were kind of saying, we can't even do this. We can't do it. It finally got done. It was not at all envisioned that we could do more than one genome in a reasonable time. And the first one cost $100 million or whatever. And we worked on it for many years. But then with the advent of next-gen sequencing, that, of course, all changed. And it changed in a way that really let us look at genetics and genomics of a large number of people, and to look at what we're really interested in, which is the variation that contributes to what we are. But it was clear that these short reads were probably missing things, particularly structural variation and rearrangements, which have been shown by a number of people to be fairly common in the genome and also important in a number of disease states. So there was, as I said, this evolution of genome references. The first references, C. elegans, yeast, Arabidopsis, which we worked on, human, which we worked on, were actually pretty high quality. C. elegans actually is completely finished with no gaps. And it's a bit over 100 million bases. But then it really became clear that while that was expensive, that finishing step was very expensive, you could do a low-quality genome that get you a lot of information for a fraction of the money, but basically whole genome shotgun, and then layering that back on the reference, so resequencing rather than sequencing. And there were also some just fairly low quality, very low quality realistically, de novo genomes done. When NextGen came along, again, NextGen sequencing, I'm speaking specifically of Illumina because that's the dominant technology, does not typically do a de novo assembly. You don't start from scratch. You map reads back to a reference. If there are structural variants in that reference, it's very possible that you will not even see it. It'll just look like mismatched reads. And so that technology is limited in looking at structural variants. The nomenclature of these sort of generations is getting absurd. They should probably just do what Apple does and call it, just call them all the same thing and let people figure it out, because it's getting silly. But what we'll call third generation sequencing is long read assemblies. And this is based on two technologies available now. When we started, there was one, which is Pacific Biosciences. We were a test site for them. Oxford Nanopore we're also a test site for. And they're both continued to develop. And so what we're doing now is using the PacBio instrument in particular. The Oxford instrument is more in a developmental stage, but to actually look at biological questions. The evolution, just for people who may not be familiar, of PacBio has been slower than they said, but has occurred. We started with them, we were getting average read lengths of about 800 bases, which was a lot bigger than Illumina, but it's 800 bases. With changes, incremental change, and yields of maybe 20 megabases per smart cell, which is their unit of their physical thing you put in the machine. Clearly, sequencing a human with those kind of numbers isn't going to happen. And so it was used for other things. However, the chemistries have evolved through several incremental steps. And starting last fall, we started testing a new chemistry, which is now commercially available, that gives us read lengths in excess of 10,000 bases and yields on a good cell in excess of 1 billion bases per cell. So that's a dramatic improvement, and it's such an improvement it really changes not only what you can target, but what you see. A 10 kilobase read is very different than an 800 base read. So, excuse me, in collaboration with Mike Schatz at Cold Spring Harbor, we decided to do a test case. And that was to sequence a cell line, SKBR3, that is a fairly extensively studied cell line containing an amplification of the HER2 gene, which occurs in a substantial number of breast cancers with a generally poor prognosis. And we also, we picked this because it had been known from even Illumina sequencing that this thing is really scrambled up. It's a, you know, genomically it's a mess, and we wanted to put the long reads to a tough test. This is a picture of the genome, and you can see that, you know, that's, you know, these things are really messed up. And in fact, what HER2 actually has done is jump from 17, which you can't read, to 8, and then it has been part of this blow up of chromosome 8, which is where the implication comes from. So we did the PacBio sequencing of this in collaboration with John McPherson's group at the Ontario Cancer Research Center, Jim Hicks also at Cold Spring Harbor, and Mike Schatz. We just used long read data, Illumina data. Initially, we mapped it back to the reference to see what it was like, but we also did a de novo assembly. You need longer reads to do de novo assemblies, and these are long enough. And then we're still doing the analysis, but basically want to use these assemblies to really understand what's going on there. In addition, which I won't talk about because we haven't done the analysis yet, Jim Hicks has done 100 single cells from this same thawing of the cell line with Illumina sequencing, with single cell sequencing. Nick Navin has also done 100 single cells or more from this. And once we get this genome put together, we're going to look at the heterogeneity in the single cells, but that's in the future. This analysis, much of it was done by Maria Natastad, a Watson School student in Mike Schatz's lab. So this is the read length distribution over the whole project, and you can see that the mean is a bit under 10 kilobases. And the problem actually we had, and I realized this early on, once the PacBio chemistry started getting much better, was that everyone that worked in my lab had started doing this stuff after next-gen sequencing. They were really used to getting 500 base fragments for Illumina, which doesn't do you any good if you can read 10,000 bases. So we had to totally change all the template preps, do pulse field gel electrophoresis for quality control rather than standard electrophoresis, do preparative gel electrophoresis for large fragments. So it was really a lot of biochemistry that had to get done to make this happen. But it was worth it. You can see this very long distribution with many reads going out to 20 or 30,000 bases. If you look at over time, and this is from Ontario, they actually, John's group actually solved this read length problem before we did. This was from November of last year, and this is incremental increases largely in the template prep and loading of the cells. The chemistry didn't change at all from PacBio. And you can see almost a doubling of read length. And again, going over 1 billion bases on the yield per cell. This shows the coverage of the chromosomes, and that's chromosome 8 that we're interested in. And you know, basically, it's fairly high coverage, distributed fairly evenly over the chromosomes, about 70x total. In looking at some regions, this is something else we're actually interested in. PacBio seems to have less GC bias, and so and some other biases. There may be better ways to do the Illumina chemistry than we did that could even that out more. But we see very even coverage over the HLA region. This is a notoriously bad region for Illumina sequencing because of the repeats and such. The long reads let us actually, we think, we're working on this, but we think it will let us actually phase through the entire HLA region, which actually is a pretty significant thing to be able to do just because of its role in a large number of diseases. One of the things that Maria did was basically to look for structural variants, and there's a variety of algorithms to do that, and she used several of them, including lumpy, localized assembler, as well as just looking for split reads. When this is done, that was from the mapping back. But you also see with the de novo assembly, with the contiguity is much higher with the PacBio than the Illumina. The number of contigs is less. The N50, which is what people use as a metric for contiguity in an assembly, is with Illumina, and so that means half the genome is represented in contigs of a size larger than that number. So if an N50 of 100 kilobases, it means half the genome is assembled in contig N50 of larger than 100 kilobases. With Illumina, the N50 was 3.3 kilobases, and with the high coverage PacBio, it was 2.17 megabases. Clearly, if you're trying to infer genes next to each other, regulatory blocks and so forth, that's a totally different product to start from with basically a 100-fold higher level of contiguity. In addition, we found indels in the assembly. The exact number we're still looking at. It depends on the filters you set, and we're validating a lot of these experimentally. But it's in excess of 10,000 indels, insertion-deletion sequences, that are present in the PacBio sequence and identified that aren't in the Illumina sequence. This is, in fact, consistent with a paper published from Evan Eichler's group in Nature and the Washington University group in January, where they did the high-coverage PacBio sequence of a haploid genome from a ididoform mole and found 26,000 indels that were not present in the Illumina sequence of that. And which some of them, looking in the Illumina data, are actually common variants in the 1,000 genome data, but had not been identified as variants because the software just couldn't pick up the indel with the short reads. These things are by split read mapping. You see there's lots of translocations, rearrangements, duplications throughout the genomes. We always show things like this, but at one level I hate graphics like this because you can't interpret it. But it does show that there's a lot of things in a qualitative way that are rearranging from chromosome to chromosome, as well as within chromosomes. In looking at the HER2 region, again, this is a very well-studied region. If you look at PacBio, you see this structure. So this is basically genomic copy here along the. So it's been normalized for copy. It's not coverage. And you see the structure when you zoom in. You see this is the region around the HER2 gene based on the PacBio sequence. If you compare that to Illumina, again, this is an 8 megabase region now on chromosome 8 that it's jumped over. This is what it looks like. And if you look at that, it looks at least very similar. And it is similar, but it's not the same. So the breakpoints identified from the PacBio sequencing are shown here, flanking some internal ones. There's an inversion here that shows up. When you analyze the Illumina data, you, in fact, miss the in breakpoints, you miss the inversion, and you add some things that probably aren't real. In looking at why that might happen, it got deleted from this slide, but basically there's a 21 base repeat that is scattered through this region, and it tends to cluster in the areas where the Illumina missed. And Illumina reads are short enough that with the 21 base repeat, it interferes with the mapping back. The translocations of HER2 go to chromosome 8, as I said before, and it shows that the HER2 gene moved from 17 to 8, and then in multiple places actually, and then continue to duplicate on 8 as chromosome 8 itself duplicated. And again, this zooms in on one of the sites, and you can see that it's different. When you look at chromosome, well, this is Illumina on both chromosome 8 and 17, showing the differences. Sorry, PacBio. That can't be good. There we go. Sorry about that. Okay. And you can map basically where the recombination points are, again, with the PacBio reads, because they're long enough to do that. And you can see there's a number of them. Some of them make gene fusions around within the region, in this case, 50 million base region. There's a number of gene fusions. One of them is here. It creates a fusion between these two genes. This was known from Illumina sequencing. And the PacBio confirmed both, and there's the other one shown here. Both of those gene fusions were previously known, and both were confirmed by the Illumina data. And then this region down here was also joined. In looking at again the region on chromosome 8 and 17, you can see the differences as the rearrangements occurred. You can map those fairly accurately. We are validating all of them with Sanger sequencing. And again, this inversion that occurred here in the smaller area. Kind of zooming in. In looking at the data in aggregate, it appears to show this following series of events happened. First of all, you start out with a healthy genome, non-cancerous. Then there was a translocation to chromosome 8 of this entire block. Then there was a duplication of part of the block and a duplication of a second section. It didn't all duplicate, and it inverted at this site, and then a final duplication here. And this was at one of the HER2 sites on chromosome 8. In looking at the transcripts, there were a number of fusion products known as I mentioned, and in fact those were identified, 7 of the 9, and some additional ones were identified, actually quite a few, about 30, that were in the process of experimentally validating that were not really seen in the Illumina sequence. One of them is shown here, where with the long reads you see very clearly this fusion project gene between the trio and the FBXL7 gene that were initially a megabase apart, but due to the rearrangements were brought together to form a gene fusion product. In addition, genes were interrupted, of course, by the various things. And this shows, again, using one of these sort of spaghetti wheels, the fairly significant number of gene fusions identified from the PacBio data that are doing reads on the long messages. So this is doing, most of these messages aren't 10 kilobases, but it's basically doing full length of what's being pulled out of the cDNA. The coverage through the area is, again, fairly even. Most of the gene fusions, many of them had been identified. What we're seeing and we're trying to quantify is that within a given gene fusion, there are, in fact, different splice isoforms that you don't see with Illumina. The isoforms being the toughest thing to find with short reads. The downside of using PacBio for transcriptome analysis is that you don't do a lot of reads. So we typically do, if we really want to look at a transcriptome, we typically do at least 100 million reads on Illumina, and you really can't get near that on PacBio right now. So that's the downside for looking for rare events, rare isoforms. You won't see them. More common isoforms, though, are much clearer to see because of the long reads that clearly transition from one exon to another that you don't get with short reads. We're working with a number of people in trying to improve that by various methods of normalization, but obviously normalization also has its limits because you're getting rid of stuff, and it may be sometimes stuff you don't want to get rid of. All the data from this project are publicly available under the terms of the Toronto Agreement, which are fairly limited restrictions, and were published in Nature. It's on, there's the address where you can download the data and do what you want with it. And then the whole genome assembly is, that is not actually available because we're still checking it. The raw data is available. And also the slides are available on Mike's website. He's much more internet adept than I am. Well, I'm not badly internet adept, but he's really a young computer scientist. So He really has been good at this. And we're continuing again the analysis on both the genomic DNA and the splice isoforms, and then the HLA region will be next. We're very excited about this because clearly, if you're missing 10,000 genomic events, it's pretty clear to me not all of those matter for much of anything. It's equally clear that some probably do in a given case. So I think we are now able to see a view of the genome with these long reads and the transcriptome that we haven't been, and it's really improved our resolution of how we can see what's going on in the genome. And as I said in my introductory remarks, that's been an ongoing process, and I think this is a fairly large leap in our understanding. And what remains to be seen is what we do with it. I mean, there's a number of options. One is, can we use these data to identify at least the more common structural rearrangements in the genome and then pull them out of the Illumina data? And Evan, as I said, did that with one of the indels in the hydidiform mole, where it was actually found to be present in 10% of Northern Europeans, but not identified. A lot of people, specifically Illumina, as you can imagine, are trying to do that. And then another challenge is, how does this technology itself develop, both in terms of the chemistry and preparing things, but even in the bioinformatics. I mean, Mike's group and others have had to redo a lot of the bioinformatic approaches because they've been developed for short reads, and they don't work efficiently with long reads. So I think those are the challenges, but the benefit is going to be huge in really understanding at a much more sophisticated and detailed level human genetic variation. And with that, I'd like to thank various people. Maria in Mike's lab, who did a lot of the analysis, Sarah in my lab, who heads our tech development group and did a lot of the development to optimize the PacBio, and then other people in our group. Karen at Ontario Cancer Research Center is the one that really figured out the most efficient way to make these libraries and helped everyone along. And DNA Nexus has helped with giving us a lot of computer time to do de novo assemblies. We have a pretty big cluster at Cold Spring Harbor, and to do a de novo assembly of a 70x PacBio genome of a human takes about a month. So they have more and bigger and faster, and they've been doing this for us. And then PacBio, who actually contributed both intellectually but also actually supported the work substantially through reagents and otherwise. Thanks. Be happy to answer any questions.
Any questions, folks?
It was interesting that, thank you for the talk. Thank you. Are you going to explore possibly doing assembly with both the long and the short reads?
We've actually done a lot of that and have published a few papers with Sergey Korin with one, Korin et al. That Mike and I were on. Yes, we are doing it. At this point, it appears that it doesn't work as well as a pure PacBio at very high coverage, and at very high coverage being the caveat, because that's $80,000 to $90,000 project for reagents, as it is. But yeah, we actually are doing that. Now that one reason, in fact, we oversampled with the PacBio is we want to then do a reconstruction downsampling. We've also sequenced this like 200x Illumina, and so we're going to do a downsampling of mixed mixed read types and see. It's computationally more challenging, but the cost benefit could be so great that, yeah.
Because I know that the Broad has been looking at PacBio Illumina assemblers, or developing those tools. Might be worthwhile to do comparison with what you guys come up with.
Yeah. I mean, we did it initially on yeast about 3 years ago, I think, and it had a few papers. But it's clearly a moving field. Yeah. Even the PacBio assembly alone is a moving field. So it may help a lot.
More questions? Okay, then thank you again.